Well, thank you so much for joining us today. My name is Jasmine Victor, and I work with Alzheimer's Orange County as the Director of Education. Alzheimer's Orange County is a independent nonprofit organization. We've been serving the community since 1982. Started by caregivers, and we're looking for to educate and support caregivers, those with dementia or Alzheimer's and related dementias throughout the community and those who support them. And today we have our special webinar. This is about a year's, I think it was a year and 3 days since last time we had Dr. Duma on to talk about his novel study here. And Dr. Duma, if it's your first time joining us here, Dr. Duma is a board-certified neurosurgeon and serves as medical director of Hoag Memorial Presbyterian Hospital's brain tumor program, and he's the chief of service of the surgical movement disorder program in Newport Beach. He has practiced neurosurgery since 1993. He's created a patented gamma knife leading edge technique to halt the migration of primary brain tumor cells throughout the brain. He's also done deep brain stimulation, FDA approved for treatment in Parkinson's disease, essential tremors. And also, he's the world's first experience of injecting fat-derived stem cells directly into the brain for treatment of neurodegenerative disorders like Alzheimer's disease, ALS, MS, and brain injuries. And so he's currently in the phase 1 trial, and he's here to give us an update on the progress with that. Before he joins, or before he goes in and into his presentation, I do want to do a little housekeeping here. Want to make sure that everybody's questions, get accounted for. But however, we'll be doing a special Q&A at the very end. So as the question comes up, feel free to type it into the Q&A. At the very bottom there, your toolbox, and we'll get to that at the very end. We won't be unmuting anybody. You'll be muted and without cameras throughout the presentation. And again, we'll just answer all the questions at the end. This is a non-CEU special presentation. We're going to be doing these monthly from now on. So thank you again for joining us for our first one. Dr. Duma, thank you so much for joining us. I love to hear all the updates that you have for us.
Oh, Jasmine, thank you. That was a wonderful introduction and well done, well said. It is true. It's been a whole year since I was here last, and time flies. I mean, it's as we get older, these years just fly by. It's ridiculous. Yeah, what I'm going to do today is I'm going to tell the community about the trial that has been FDA approved, FDA cleared. I can't use FDA approved. FDA cleared to actually use stem cells for the first time, injecting the cells directly into the brain. So let me pull up my slideshow here. Let's get this going and make sure that we're doing this right. Can you see the whole screen?
Yes, thank you.
Great. Okay. So yeah, this is what we discussed a year ago, literally, because we got this clearance by the FDA 1 year ago. But when you get FDA clearance, then you have to get a sponsoring hospital. You have to get approval from the IRB to do it, the local institutional review board. There's all sorts of processes that go through, and I'm going to take you through that as kind of a fun journey that we took just to get this phase 1 approval. But let me tell you a little bit about the phase 1 approval. I mean, the title says it all. We are the first in human to inject cells directly into the brain for Alzheimer's disease. And I'm going to assure you that this is probably going to become a very important route of treating brain diseases. And we're not just talking about Alzheimer's disease, we're talking about ALS, we're talking about MS, Parkinson's, stroke, traumatic brain injury. This has a lot of legs, a lot of things that we can do with these cells to treat different disorders that are similar to Alzheimer's disease. But the very first in human is going to be for Alzheimer's disease. I'm happy to report for this crowd. Let's go over the inclusion criteria. So every single time FDA approves a trial, it becomes uh, uh, you have to satisfy various what they call inclusion criteria. So it's not open to the general public. It's not open to anybody. And if you do have Alzheimer's disease, you have to satisfy a criteria as well. For example, the first criteria is that you have to actually have a diagnosis of Alzheimer's disease. We can't have, frontotemporal dementia or Lewy body dementia or something like that. They want you to have this diagnosis of Alzheimer's disease. The patients have to be less than 80 years old. Uh, and you have to be a fast stage 4 and 5. Now, if you go to your neurologist and they tell you what stage you're in, the stages are over here on the right. This is a little staging system for Alzheimer's disease. And you see that, stage 1 is a normal patient. Stage 3 is called really mild cognitive impairment. Stage 7 is very severe. Now, we're treating the stages 4 and 5. So if you don't know what stage you're in or your loved one is in, you have to find that out, because if you're a stage 3, you can't be enrolled in our trial. If you're a stage 6 or 7, you can't be enrolled in our trial. Those will come later. Those will come later in our plan with the FDA. You also have to have a mini mental status score between 10 and 20. The highest MMSE is 30. So it's a test that is very quickly given, and I would imagine every patient who's under suspicion for having Alzheimer's disease will undergo this test. It is about a 20-minute exam. It's a written exam as well as an exam that asks you questions and verbal. There's some verbal parts, there's memory parts to it, there's drawing skills parts to it, etc. So you have to have a score between 10 and 20. Remember, 30 is the highest. So we're looking for patients within this middle range. Finally, you can't have any other major comorbidities, like you also have cancer, or you have active heart disease, or something dangerous for you during the course of this trial. We like you to have a spinal fluid study. That's a spinal tap where we send the fluid to see if there's beta amyloid or p-tau, phospho-tau levels in that fluid. We'd also like you to have had a brain PET scan. That's either a regular PET scan, a glucose PET scan, or an actual beta amyloid PET scan. And then if you fit in these criteria, either you or family members can do this. And what I suggest right now is if people who are on the screen can take a photograph of this screen right now with their iPhones or a screenshot, and that way you have our phone number. I'm going to show this again at the end, but you have our phone number as to how to reach us if you want to become part of this trial. I mean, this is a very exciting time, and you would probably want to become part of this trial, especially if you were a stage 4 or 5 patient. Even the new drug Leqembi is not indicated for stages 4 and 5 yet. It's only for stage 3, which I'm going to go into later on. But do me a favor and screenshot this slide or take a photograph with your phone so that you know who to reach. I will show it again, okay? Uh, and now we'll just move on into the talk. All right. So, as Jasmine alluded to, I am the director of the Brain Tumor Program, and I'm chief of service of the Functional and Movement Disorder Program. So, what does that mean? Why is a brain tumor guy doing this? And why is a functional movement disorder program guy doing this? Well, functional neurosurgery is a subspecialty, which means you deal with the functional aspects of the human, such as movement, such as thinking, cognition, etc. So that's where I fit in this regard, and that's what kind of propelled me to start treating Alzheimer's disease and others with stem cells. I've founded a number of companies, and I've also invented a number of things that have United States patents on them. And the most exciting one recently is the one I'm going to describe to you today. The problem of Alzheimer's disease is obvious to all of you in the audience. There are close to 7 million people in the United States alone. 1 in 3 seniors will die with some form of dementia. ALS is a big problem, multiple sclerosis, Parkinson's. These are all problems, people, that right now have no fantastic treatments. You know, the treatment of Alzheimer's disease has changed a bit recently thanks to Leqembi being approved. But before Leqembi and aducanumab, it did not have anything for 20 years. There wasn't one new drug for 20 years treating Alzheimer's disease. There have been combination drugs that came out, but things like that, there have been nothing novel. And what I'm going to show you today is super novel. Some of the statistics, again, this is straight out of the Alzheimer's Association website, but it's mind-boggling as to what this could entail going forward. It's going to cost the nation, 345 billion into this year. By 2050, we believe that the costs of treating patients with dementia may go up into the trillion range. And we're going to need a lot more workers. That would be people taking care of patients directly or indirectly with Alzheimer's disease, needed to care for the growing population. So this is going to be a very big problem going forward to even get enough manpower or people power to do it. Now, what is wrong with what's been going on? Well, there's two things. One is the blood-brain barrier. You've all heard of that. And the other is that these diseases, including Alzheimer's disease, has all sorts of problems related to it. And, here are just a number of things going on in Alzheimer's disease that you can't even read them, some of them. It's like a different language, but just take my word for it. All of these things are happening in the brain of an Alzheimer's patient. And if you target one of them, like let's say you target neuroinflammation, it may not fix the other problems at the same time. So that's what got me going with stem cells. The drug that exists right now, Leqembi, is a Biogen and an Eisai drug, Eisei. That's another drug firm from Japan, or I believe it's Japan, and they have been working together to make new drugs that fight the diseases. Now, you may remember this drug Aduhelm that came out back about a little more than a year ago, and that really got deep. That was out. It just didn't work very well. The European Union wouldn't accept it for their patients because it was so ineffective and it had a lot of side effects. Well, they then, the same concept came about with Leqembi, which is lecanemab, and that just got approved. and is approved for certain patients only. And the main problem with these drugs, and this is your take-home message for the big pharma work that's being done, is this. And I want this funny cartoon emblazoned in you. Uh, Imagine if at home you had a refrigerator and it was broken and it wasn't working and your food was spoiling like this on the right. Well, of course, you could keep taking out the spoiled food and throwing it in the trash, and you can put more food in the refrigerator, but it's just going to spoil again. So the concept here is you have to fix the refrigerator. And the analogy is the refrigerator is the Alzheimer's brain. The the food is basically amyloid beta and p-tau. So what amyloid beta and p-tau are they are the end products of cell death. So your cells in your brain have already died. They crinkle up into little tangles and plaques. They sit there in your brain, but are they themselves the real problem? Or is it the fact that the dying cells before that are the real problem? So what you want to do is, sure, you want to clear out the debris, right? Or the spoiled food, but you need to fix the refrigerator. And in my mind, my simple brain surgeon mind, the only thing that might be able to do that is stem cells. And that's why I went that direction. So I'm going to give you this little history tour of how I started in 2014. So we're talking 9 years ago. I had this idea that if we injected stem cells in the brain, maybe it would do something. And I was targeting all these diseases. I wasn't just targeting Alzheimer's disease. I was targeting all of these neurodegenerative diseases that still don't have any cure or major treatment. For example, we don't have a real treatment for traumatic encephalopathy, which is what football players get from their combative injuries. Um, I'm a neurosurgeon, I'm a brain surgeon, so I deal with the brain every day. And I thought the most desirable way to deliver these stem cells would be to deliver it into the center of the brain, these little hollow cavities. Now, I bet you all in the audience don't know that our brains are hollow. Everybody in the audience has a hollow brain, and that's not an insult, it's just real. Our brains are like a honeydew melon. You cut the melon in half, it's hollow in the middle. And these are the hollow spaces. They're called ventricles. And these are the spaces that produce the cerebrospinal fluid. This is the fluid that bathes our brains. And if you took out the sample, you can get a sample through a spinal tap. People know about spinal taps, but they don't often realize that out of a spinal tap, the fluid is crystal clear. It looks like spring water. And this fluid, this water, washes our brains and spinal cord every day on a regular basis. We actually produce the fluid about half a quart of fluid per day. And then that washes out the brain. It clears out the brain. It acts as a waste disposal system. And our brains are literally floating in this fluid as well. So wouldn't that be a great place to inject the stem cells right in the middle and then let the stem cells percolate into the brain itself and fix everything magically that we have going bad with us? Okay, so that's the concept. And in order for me to do this back in 2015, I went through what's called an institutional review board, called an IRB. And they said, well, why don't you prove it to us and show us that this works in some way? Let's do some animal work. Show us some animal work. So we did. We operated on the rat, and we put the little cells into the ventricle. And this is proof here that we were able to get in the hollow cavities of a rat. And this is proof, you can't see this, but this is proof right here that the cells percolated into the brain itself through the ventricles. So we were able to tell the IRB, well, yes, I think the cells get in. We need to see how that affects the people. And at that particular point, we got approved to do some pre-FDA work. Now, this is a preview to the FDA work that I'm going to tell you to get enrolled in. So the preview is as follows. We were going to inject cells every 2 months. And remember, this started in 2015. How do we inject the cells? Well, you inject them through a little port that is placed under the scalp and delivers the cells into the ventricle. And you could pierce this with a needle and inject the cells with a needle. This is not new. I did not invent this device. It's called an Ommaya reservoir, and we use it in the brain for chemotherapy. Are you all familiar with a port that cancer patients can get that goes into their large vein? That's called a portacath. To put this in is 3 times safer than putting in a portacath, believe it or not. This is a very safe procedure, putting this into the brain, and it takes 15 minutes. So it's a very quick procedure to install that. And once that's installed, we can inject it with a needle, okay, through the scalp. It's under the scalp. It's not visible. And here it is. Here's the real picture. So the object here would be to get some stem cells from the fat of the abdomen. So adipose-derived stem cells, ADSCs, adipose-derived stem cells, spin them down, and then inject them directly into that port. And here is an actual patient with a port, and all you do is you have this at the bedside. This is betadine. You wash their scalp, you draw off some fluid, and then you inject the stem cells. And this is what I did for 5 years. And that's a close-up. So this is a very painless and very simple procedure. The act of injecting the stem cells takes no more than 30 seconds. As a comparison, if you are a Leqembi patient, the new drug, you have to get an IV put in, and you have to have this IV done every 2 weeks. Whereas our study, this would be done every 2 months. And I can assure you that putting an IV in and infusing that drug is far more morbid than what's happening here for this gentleman. Remember that staging system where we did in our pre-FDA work stages 4 and 5 patients. Now, this curve is very important. This is the Alzheimer's disease curve. Every Alzheimer's patient follows this curve in one way or another. When you get diagnosed with Alzheimer's, you are scoring on that mini mental status score. Remember, I told you we were only enrolling people between 10 and 20. So that would mean this part of the curve right here. So the people on this part of the curve are who were in our pre-FDA trial. And you can see that's a slippery slope. It's a very rapid decline that usually occurs over a couple of years. And people who know Alzheimer's patients or have Alzheimer's patients in their family know about this curve. And the interesting thing is that if there were any drug that changed the shape of this curve, that would be fantastic. That would be earth-shattering. Right now, Leqembi only takes this end of the curve and pulls it out by 5 months. It doesn't change the shape of the curve, and it just makes the patients have longer quality of life for 5 years. No, excuse me, for 5 months. So that's not a huge change. But what we really need to do is change the curve, right? So we saw in our pre-FDA work, so now we are now injecting patients with their own stem cells that have not been altered in any way. And what we saw is an inflection point. We saw a change in this curve. This is the normal curve up here of decline of an Alzheimer's patient. And this is over the same timetable. This patient was diagnosed up here with Alzheimer's disease. We gave them their first injection of the stem cells, and lo and behold, they stabilized. Well, let me just explain. This is the first time this has ever been seen in the field of medicine. Well, this could be a one-off. This could be just one patient. Well, we looked at them longer. So this patient went out 2 years with stability. And this is monumental in my opinion. We also saw p-tau levels go down, and p-tau is the bad marker for those tangles and all. And then amyloid went the proper direction here. This is an index upward, an uptrend in this is better too. So we saw this in our patients, and it got me very excited. We also looked at other patients, and this is for Alzheimer's patients. For 4 patients, we saw stabilization of the memory performance index, which is like the MMSE. We saw this in 4 patients stabilizing and followed them more than 4 months. So this was very exciting, very optimistic for what we might be able to do for Alzheimer's disease. That's never been done before. We then, in one patient, we actually saw growth of the hippocampus. So you guys know what the hippocampus is. It's the short-term memory area of the brain, and it's responsible for your short-term memory. So if everybody complains, oh, my loved one can remember everything from their childhood, but they can't remember anything that just happened, and then they repeat themselves. Well, this is that area. And if you look at a scan of an Alzheimer's patient, this little area, the size of your pinky, the size of your small finger, is shriveled up like a raisin. It's not full and happy anymore. And what we saw in one patient is we actually saw it grow to a relatively normal size, to a 50th percentile, like an average size. So that was exciting also. And this is the actual imaging that is done. Artificial intelligence actually measures the volume of the hippocampus and sees whether it is growing or getting smaller. And in this case, it went from less than 5th percentile for age to the 51st percentile. So this was exciting also. Now here's the cerebrospinal fluid test. We tested, remember I told you p-tau is really a big thing that we test. And when p-tau is way out here, that is bad. Okay, you are considered to have Alzheimer's disease. So any movement of this asterisk would, in this direction, would be beneficial, lowering the amount of p-tau. So we had one patient where it went from here to here to here over a period of 8 months. This has never been seen before in the field of medicine. So this was exciting as well. And we felt that it had to be due to the effects of the stem cells. Now let's switch gears here. I want to show you a video. This is a young lady with multiple sclerosis. Okay, I know this is an Alzheimer's group, but bear with me. This is a very powerful video showing the effect on a multiple sclerosis patient. This patient came to us with 10-year history of multiple sclerosis, and she was unable to walk more than 1-inch steps. Check this out. So this is the way she came in to us. walking only inches at a step. So she came to us because we were offering stem cells. And she went and got her little reservoir put in her skull. And then 4 months later, look at her. She's walking a little better. Now, she's on no other drugs, no other medications for MS. And just 4 months after the first injection, she's beginning to walk like this. Now, this might be fine. This might be another one-off where maybe this is just a fluke. This is her 14 months later. And she has the progressive form of MS, which means that if she were to have stayed where she was when she came to us, she should be worse. She should be in a wheelchair by now, but she's now walking. 2 years later, this is her. She's walking. She's, believe it or not, driving a car now and far more fully functional than she was when she came in. And remember, it's been 2 years. So she should be, at this point, she should be in a wheelchair or have passed away. So what happened was she got a phone call from her people at USC to try a new drug. And the new drug was, it required that she get out off of our stem cell work and that she try the new drug, and she'd have to do the drug for a year. And after a year, she came back to our office, and this is what she looked like. So remember, she had been off of our stem cells for 1 year, and she's unable to take one step. So to me, this was a very powerful experience. She had gone so far in those 2 years, able to walk, drive a car, walk independently from taking 1-inch steps. And now she was off of our stem cells and is completely unable to walk. So this made me publish our work in the medical literature. This is a peer-reviewed journal, well-respected journal, and it has been published. It was published in 2019, and it is the first published paper of its kind. And it's a placeholder because we're the first in the world to do this. So to summarize, we injected a total of 39 patients over 5 years using IRB clearance in these various diseases. And we used their own cells. We didn't modify them in any way, and we didn't grow them in the laboratory. So that was wonderful. But what that really did, especially with the results that we had, and essentially less than a 1% complication rate, it propelled us to work with the FDA, create our dream pharmaceutical and our dream corporate team. So we formed Regeneration Biomedical. To do an FDA trial is extremely expensive. And I'm not joking. I mean, Regeneration Biomedical has already been through $6 million of investor money to get to where we are. And we're moving forward. We have a $10 million round right now. So this takes money to do, as you might imagine. And no one blinks an eye when Big Pharma does something because they've got billions of dollars behind them. But if you have a smaller company like ours, this is how we do it. So Regeneration Biomedical was born. This is our corporate team. Some of you in the audience may know Hans Kierstead. He's the one that started California STEM, which was our UCI's stem cell program. And he's a professor at UCI, and he is our scientific advisor for this. We have a excellent staff of people working for us, and we use the laboratory, which is Hans Kierstead's laboratory in right here on Von Karman in Irvine. So it's very close, and we're very active with them. Our product, we've given a name called Cerawint. And what exactly is that? Well, our dream pharmaceutical would be, wouldn't it be great if we were giving a million cells before the FDA protocol? Wouldn't it be great if we gave 20 million cells? And that's what's called expanded. So you grow the cells in the laboratory. And what if we were able to supercharge them with something called Wnt? Wnt is an activating substance. It is a protein at the that stem cells have, that when that is expressed, it turns on other stem cells. So in other words, I liken it to an on-off switch. So if your stem cells are expressing Wnt, then your stem cells are being stimulated, and other stem cells that are laying dormant are being stimulated. And by the way, we all have stem cells in our brain right now. Everyone on this Zoom has stem cells in their brain that are just pretty much laying dormant. And it's our hypothesis that by squirting stem cells into the ventricles of the brain, we may be waking those dormant stem cells up. So that was our theory. That was our hypothesis. We submitted it to the FDA in 2020. And you want to know something? 27% of the trials submitted to the FDA are accepted. That means 73% that the FDA sees, they say, we don't want any part of it. Forget it. We were in the 23%, excuse me, 27%. So we were accepted by the FDA to move forward, working with them to get them finally to give us the clearance for a phase 1 trial. So we worked with them for about 16 months. You see, March 2020 to June 2021. And finally, they said, look, we like what you're doing, but it's different from what you did before, and we want you to inject the cells into a large mammal. A large mammal is usually a monkey or a pig or a dog. We went with pigs. So this is a pig. That's me drawing out the area surgically that I was going to implant the reservoir. And then this is the pig after they were operated on and they had a little reservoir put in. We actually did this pig work down in San Diego. The cost of this was close to $1 million just doing the pig work. And the result was the pigs are sacrificed, their organs are harvested, and they get sent to Indiana, where they look at everything and everything was perfect. So in other words, the stem cells that we injected into their brains were not circulating into their heart, their liver, their lung. They stayed in the brain, and there were no adverse effects. There was even no inflammation per se. So after that, we got this letter from the FDA, which was to remove the clinical hold, meaning we're allowed to do a phase 1 trial in humans with FDA clearance. There's a lot of champagne was poured this night, and a very important and very, very monumental day for stem cell research. We also got a patent. When we started this work, 3 years ago, we submitted for a United States patent, and you see the picture of the reservoir. This whole system is patented to put stem cells through. So we got it and we own it. Let's just compare the stem cell work that we did before what we're proposing in the phase 1 trial, comparing to that of the results of Leqembi. So remember I told you we chose the worst stages than they did. They were just doing stage 3 patients in their study. Their results showed cognitive impairment slowing, just slowing, and they only saw it in 27% of people. So what you're seeing here is that a FDA approved a drug to be on the market for Medicare to use that only helped 27% of the people. I mean, that's how, you know, that's how bad we needed a drug for Alzheimer's disease. Our drug in all of our Alzheimer's patients showed some sort of improvement or stability in 90% of patients. Risks and complications of Leqembi, I don't know whether you read it in the newspaper, but 13% of patients get brain swelling from it, and 17% get brain hemorrhages. We saw no brain swelling, we saw no brain hemorrhages, no deaths, and it makes sense. We're using the patient's own stem cells, so we don't expect anything bad to come of it. So it's very exciting to see our results compared to Leqembi's results, and then to see how this goes forward. And the only way to see that go forward is with a phase 1 trial with the FDA. And this is now where we're at. I'm bringing you up to date. August 14th of this year, so a few months ago, Hoag did all their diligence. Hoag Hospital did all their diligence. They went through the IRB that we use. They went through the legal department. They went through the informed consent and everything, and they finally gave us the approval to do this trial at Hoag. We could have used any other hospital. I could have gone to Mass General. I could have gone to MD Anderson. We could have gone to Harvard to do this trial. But, I am a little bit biased, and I love Hoag. I've been at Hoag now for 26 years. So it's just a wonderful institution. And they have just gotten a lot of financial support from the Pickup family for cognitive care. So they're very in tune with all of this. And so they just approved it. Dr. Gus Alva, some of you may know him. He's an Alzheimer's specialist as well. Gus Alva is our principal investigator. So he oversees the trial now. It is listed in your trial match in the Alzheimer's Association. Trial match, you probably know, you type in various keywords and you can find, so if you typed in stem cell and Alzheimer's disease, you'd find our study, and there it is right there. And then it is also on clinicaltrials.gov, so the government's website, the government's own website for the clinical trials around, and you can see it right here, and you can see the word recruiting, and that's exactly where we're at. So we are recruiting patients, and all we need, everybody, pay attention, is 9 patients for the phase 1 trial. So if you want to get into this trial, we already have a list of patients, but there are still some openings. I want you to contact Regeneration Biomedical, and then we'll get you in touch with the research department at Hoag. I will give you that information again at the end of this webinar. So it is listed in the government website, and it is listed on the Alzheimer's website. We are planning on completing the phase 1 by the middle of 2024, but at the worst at the end of 2024. We're hoping that when the FDA realizes we're not having much in the way of side effects or complications, which I'm fairly certain we will succeed with because of my previous work, if everything is good, they may approve us to move forward with the phase 2 trial. And phase 2 differs in that we're looking for the efficacy. We're looking for the drug to actually have an effect. We already have, from my previous work, some clues that there is some effect of these cells that is very positive, but it is not an FDA controlled trial. Furthermore, a phase 2 trial will involve a number of other institutions. We're planning 4 different sites where we would actually have them do them. I mean, I have one in Seattle, one in Florida, one in New York that I'm thinking of using. So that would be very exciting, and that would take about 2 years. But those of you who get enrolled in the phase 1 have a real up on that. I mean, you could get an injection within a month or within 2 months of the first part of the trial. Very exciting. So it's very, tempting to get involved in this trial. This is our group. This is Regeneration Biomedical. That's Hans Kierstead on the right. And I'm going to leave, this is the last slide. I'm going to leave this up again for you all to photograph. So you know how to reach us if you have a loved one or if you yourself have the disease and you want to get involved in our trial. So Jasmine, that's the end of it. I was just wondering whether you wanted to have any questions.
Thank you so much, Dr. Duma. Yes, that was wonderful. Um, I do have a couple questions. It looks like somebody mentioned, oh, regarding the trial, do you have to live in California in order to be part of this trial?
Yeah. I will tell you that it is it the answer is no, you don't. You can definitely submit. It's going to make it difficult because you have to travel to be examined by our local doctors here, and certain tests have to be done here. But that is certainly not out of the question, and it's only every 2 months or so that we're getting some sort of testing. So the answer is no, you don't have to live here locally.
Okay, good, good. Another question. Not sure if you can answer this one. So every person who has a loved one that does not fit the trial parameters would like to try stem cells. What do we do?
Yeah, great. It's actually, I can answer that. It's a great question. So there is a situation where we finish phase 1, right? And a patient is outside of the criteria you see in front of you. Let's say they're 81 and they're a stage 4. They're not that bad, but they want to have the cells. We can actually submit for compassionate use. We can submit to the FDA the possibility of using our cells for them. And I've done that already in brain tumors. I've been involved in a number of FDA approved trials for brain tumors. And we have, on rare occasions, enrolled patients that are outside the study closes or they're outside of the criteria, and we can actually submit for that. So the answer is possibly. The other thing is, if someone has ALS, could we actually start using the cells for ALS after the phase 1 trial? And the answer is, in the same way, we can submit for compassionate use.
That was another question is, are these parameters absolute, like the less than 80 years old? So
Mm.
I thank
They
you.
are they are 100% absolute. Yeah, we have to abide by various criteria here, and if we waver, we risk we risk the wrath of the FDA. So we're going to comply with everything.
All right. If you have a pacemaker, can you still be in the trial?
Yes, I believe so. Yes, we'd have to get cardiac clearance. The only surgical procedure is the implantation of the reservoir, right? So when we put that little reservoir in the brain, that's a 15-minute procedure done almost under local anesthesia, a little bit of inhalational anesthesia, but it is very benign and most cardiologists will approve it.
Okay. Let's see. Have you done any brain imaging pre and post implant to see changes? And where do you see changes besides the hippocampus?
Great question. We have not done many in the pre-FDA trial, but we will be doing a lot during the FDA trial. But that was just one example where we measured the size of the hippocampus. That's called a neuroquant study. It is a regular MRI. You don't have to do anything special. It's an MRI, but it's called a neuroquant analysis, and we plan on doing that in our phase one. So we didn't see a lot. And by the way, you can't diagnose Alzheimer's disease with an MRI. It's, unfortunately, that is the case. You can get clues to it by a shrinking hippocampus or atrophy of the brain. You can diagnose frontotemporal dementia by looking at atrophy of the frontal and temporal lobes, but you can't make a diagnosis of Alzheimer's disease on a scan.
Right. So why only Alzheimer's and not other dementias?
Well, that's because for the phase one, we had to focus on one. Okay. So we focused on Alzheimer's disease because the others, first of all, are less common. And secondly, they share different aspects of each other. And we wanted to make it as pure as possible for the FDA. I promise you, going forward, we will include other dementias. But
Great.
that's after the phase one. You realize that if we do a phase one trial and it appears safe in the brain, it appears that this technique is safe and can move forward, we can use the same cells for Alzheimer's disease as well as ALS, MS, Parkinson's disease, et cetera. So our company plans on moving forward with all of those diseases. Maybe it'll work perfectly in MS, but it won't work in ALS, for example. Maybe. We'll see. But that will be the future going forward. In the same way, we would do subsets of dementias. So we would probably have a subset of Lewy body dementia, a subset with frontotemporal, et cetera, et cetera. So the answer is yes, it will be done in the future.
Okay. So regarding your phases, if someone gets into phase one, could they continue to phase two? But it sounds like they're different levels or different criterias for each phase. Is that correct?
No, that's a great question, and I just didn't bring it up I didn't have the time to do it, but um, understand that phase one is just one injection. Okay, so they're going to get stem cells once, and then they have to wait probably close to 6 months before they might move into the phase 2A trial. So those patients who are in phase one will get preference to move into the phase two, but they will definitely move into phase two. Okay. So the answer is if you enroll in the phase one and you're already a phase one patient, you will get into a phase two, but you will be called a phase 2A because you're not going to be the real phase two, which is the patients that didn't get the trial of the phase one, right? So the answer is yes, we'll be able to include all.
Okay. Um, so would this is this a treatment or is this a cure? So would I need to take these injections for the rest of my life?
Okay, so you saw that video, that very powerful video of the young lady who had multiple sclerosis. We kept injecting her every 2 to 3 months, even every 4 months, and she was getting better and better. We stopped the treatments for 1 year because she decided to do another trial at SC, and she came back and she had completely reverted back to almost worse than she was when she came to see us. So, and it has always been my philosophy that, people go to Florida, they go to Mexico, they go to Germany, they go to China for stem cell injections, and they get one injection. Do you really think that that one injection of stem cells is really going to do the trick? I don't think so. I think it's like a vaccine. You need to get boosters of this. And that's just simple common sense to me. So that's why the design for the phase 2 is to inject this every 2 months. So the answer is you're going to be getting an injection every 2 months. If we find that patients are doing so well with that, we'll make another subset of patients that'll get it every 4 months. Maybe you only need injections for every 2 months for a year, and then you can get a booster every 6 months. That all remains to be determined. I mean, that's the future of this.
Great, great. Well, thank you so much. Um, and let's see, just a couple, I've already answered one is that basically, again, this is a recording. We were recording this and it's going to be posted online probably in the next few days. Also, another question is whether or not we can distribute these slides. Um, Dr. Duma,
Well,
is
no, I'd rather not distribute the slides, but you can certainly post the webinar.
okay?
Absolutely. You can compare it to the last webinar that I did a year ago, which is also on your website.
Yes, yes. Again, thank you so much. It looks like we're just about wrapping up. I just want to do a couple announcements. I just want to again thank you, Dr. Duma, for all your research, for telling us about your research, sharing everything with us. And this update is just wonderful news. I'm so grateful to hear this. Thank you so much. And also, again, just a little announcement. I kind of alluded that this is a special webinar. There is no CEUs available, and we are going to be doing our monthly CEU webinars. But we're also doing an additional hot topic, if you will, special webinars once a month. And they're just kind of random, but our next one is going to be January 24th, and this one's going to be on dementia and gun safety. So it's titled Help, My Friend Has Dementia and a Gun. So thank you so much. Please join us. You can always check us out on our calendar. It is online, and I will post that in the chat as well. There you are. And thank you so much again.
Thank you,
Have
Jasmine. I very much appreciate it. Please contact us as needed for your loved ones, and we look forward to enrolling them in the protocol.
great. Goodbye, everybody.